Okay, for question 4a, how does the innate immune system detect against virus and protect against viral spread? Okay, when a virus invades human tissue and enters our circulation, they will eventually encounter your macrophages and your dendritic cells. And on these macrophages and dendritic cells, you're going to find membrane-bound toll-like receptors. These membrane-bound toll-like receptors are capable of recognizing viral capsid proteins. We also have endogenous toll-like receptors which play a role once the viruses have been phagocytosed by the dendritic cells and macrophages. Because endogenous toll-like receptors, mainly toll-like receptors 3 and toll-like receptors 7, detect or recognize single-stranded RNA and double-stranded RNA. Endogenous toll-like receptors also do not form dimers, while membrane toll-like receptors do form dimers. So what happens once membrane toll-like receptors and endogenous toll-like receptors have detected against our virus? Well, that will lead to the activation of transcription factors, mainly nuclear factor kappa beta and interferon regulatory factor. As we can see, nuclear factor kappa beta leads to the synthesis of tissue necrosis factor alpha, which I won't be covering. We'll be looking at what interferon regulatory factor leads to. Well, interferon regulatory factor leads to the formation of interferon alpha and interferon beta, which are your type 1 interferons. Once these interferons are synthesized and exit the macrophages and dendritic cells, they will attach to interferon receptors on adjacent cell, leading to an antiviral state. And what is an antiviral state? Well, its two major characteristics are mRNA degradation and inhibition of protein synthesis initiation complexes. mRNA degradation is going to be caused by the activation of the enzyme oligo-A synthetase, which catalyzes formation of oligo-A synthase. Oligo-A synthase will then activate RNase, which breaks down mRNA. As for inhibition of protein synthesis initiation complexes, this is going to be inhibited by the activation of protein kinase C. For the next question, outline how cytotoxic T cells and natural killer cells kill target cells and how MHC regulates the activation in different ways. Right. So, when a virus infects our cell, we get an infected cell. Okay. But once the virus is in the cell has proteases which break down the viruses and mount the degraded viruses onto MHC1s, which is then displayed onto the infected cell membrane. These MHC1s with the degraded virus can then be detected by T cell receptors, by cytotoxic T cells, along with CD8 core receptors. Okay, with the help of CD8 core receptors and T cell receptors, T cells can recognize the peptide on the MHC1 and recognize whether it's a self or non-self antigen. If it is a self antigen, the T cell will leave the cell alone. But if it's a non-self antigen, the T cell will release perforins and granzymes that will lead to cell apoptosis. But viruses are smart, okay, because viruses, they downregulate MHC1. When they downregulate MHC1, T cells cannot function. That's where natural killer cells come in. Natural killer cells are also part of our innate immune system. So, how do natural killer cells work? Well, they work on the premise of two major receptors, our activatory and inhibitory receptors. The main concept to understand is that when an inhibitory receptor is bound to MHC1, apoptosis will not take place. But if an inhibitory receptor is not bound, apoptosis will take place. So, during downregulation of MHC1, an inhibitory receptor will not bind to anything. Hence, you're going to have apoptosis. So, you're not going to have this. So, you're not going to have an inhibitory signal. You're only going to have an activatory signal. Hence, perforins and granzymes are going to be released, leading to the apoptosis of an infected cell. Next, for the mechanism of action of acyclovir. Well, what is acyclovir? Well, it is a guanosine analog involved in herpes simplex virus. It's a very effective against herpes simplex virus. Why? Because herpes simplex virus has an enzyme, thymidine kinase, which has a very high affinity for acyclovir and catalyzes the reaction of acyclovir, forming acyclovir monophosphate, which then forms acyclovir diphosphate via cellular kinase, which then forms acyclovir triphosphate, which is your active form of the drug, acyclovir triphosphate. What is acyclovir triphosphate? Well, it's an acycloguanosine. What it does is it incorporates itself into the growing DNA chain during viral replication. When it incorporates itself into the growing DNA chain, further nucleotides cannot be added to the growing DNA chain. Further nucleotides cannot be added to the growing DNA chain. Why? Because acyclovir triphosphate does not have a 3-hydroxyl group, and as we know, we can only add nucleotides to a 3' end. Hence, when acyclovir triphosphate has been incorporated into the growing DNA chain, we are not going to have viral replication.